Hi, I'm Kylie Savage. I do biology A-level, and today we're going to be doing a super fun video on the light-dependent stage of photosynthesis. Now, by the end of this video, you should be able to outline how light energy is converted to chemical energy in the light-dependent stage, and also explain the role of water in the light-dependent stage. I've done this video before. Stupid computer deleted it, so now we're doing another one. So to start off with, the light-dependent stage of photosynthesis takes place on the thylakoid membranes of the chloroplasts. Now, the photosystems are embedded in the membranes, and in the photosystems are the photosynthetic pigments. Photosystem 1 occurs mainly on the intergranal lamellae, and photosystem 2 only, almost, occurs exclusively on the granal lamellae. And now these pigments trap light energy, and it can be converted to chemical energy in the form of ATP. So instead we're going to go over the role of water, and the role of water is pretty important in this. Photosystem 2 contains an enzyme, and this enzyme, in the presence of light, can split water into protons, electrons and oxygen, and this is called photolysis. Some of the oxygen produced in this way is used by the plant for aerobic respiration, but lots diffuses out of the leaves through the stomata and into the air. Water is a source of, first of all, it's got hydrogen ions, and these are used in chemiosmosis to produce ATP. These protons are then accepted by a coenzyme NADP, which becomes reduced NADP. And this can be used during the light-independent stage to reduce carbon dioxide and to produce organic molecules. There's also electrons, and these electrons replace the ones that are lost by the oxidised chlorophyll. Water is one of the main raw materials used in photosynthesis. It keeps plant cells turgid, which enables them to function, and the byproduct of photosynthesis, oxygen, comes from water. So now we're going to move on to photophosphorylation. Light's got a dual nature, and we think of it as travelling in waves. However, we can also think of it as travelling in particles that we call photons. Not protons, photons with an H. Photophosphorylation, photons. When a photon hits a chlorophyll molecule, the energy of the photon is transferred to two electrons, and they become, well, we can use the term excited for it. I think this is quite a fun term to use because you just think of like the energy going to the electrons, and then they're like two excitable children. They're like, yay! They've got all this energy, so it's a really good way to remember it. Even if it is the actual term, it really helps. Now, these excited electrons are captured by electron acceptors and pass through a series of electron carriers embedded in the thylakoid membrane. If you want to remember that, think of it as you've got your really excited children, really excited little electrons running around. One of the parents is like, oh God, I've got to accept that they're going mad. So they're captured by them, and then they're like passed from adult to look after until they've sort of run out of their energy. The electron carriers are proteins that contain iron atoms. Energy is then released as electrons pass along the chain of electron carriers. So think of the energy going down as they entertain each adult at a time. This pumps protons across the thylakoid membrane into the thylakoid space where they accumulate. Sorry, I can't say the word. Don't pick up on it. I can't say the word. Accumulate. Anyway, a proton gradient is formed across the thylakoid membrane, and the protons flow down their gradient through channels associated with the ATP synthase enzymes. So, proton gradient, because of this, is formed across the thylakoid membrane. The protons flow down the gradient, high to low, through channels associated with ATP synthase enzymes. Now, this flow of protons is called chemiosmosis, and it produces a force that joins ADP and PI to make ATP. This kinetic energy from the proton flow is converted to chemical energy in the ATP molecules, and this is used in the light-independent stage of photosynthesis. The making of ATP using light energy is called photophosphorylation. There are two types, cyclic and non-cyclic. So, we've looked at photophosphorylation as a whole, but now we have to look at the two specific types. As I said, cyclic and non-cyclic. Start off with cyclic. It only uses photosystem 1. The excited electron passed to an electron acceptor and back to the chlorophyll molecule from which they were lost. There's no photolysis of water and no generation of reduced NADP, but a little amount of ATP is made. This may be used in the light-independent reaction of photosynthesis, or it could be used in the guard cells, because their chloroplasts only contain photosystem 1, to bring in potassium ions. This lowers the water potential and causes water to follow by osmosis, and this causes the guard cells to swell and opens the stomata. So that's cyclic phosphorylation. Non-cyclic is different. Whereas cyclic only involves photosystem 1, non-cyclic photophosphorylation involves both systems, photosystem 1 and 2. The first thing that happens in non-cyclic photophosphorylation is light strikes photosystem 2. This excites a pair of electrons that leave the chlorophyll molecule from the primary pigment reaction centre. The second thing that happens is that the electrons pass along a chain of electron characters, and the energy released is used to synthesize ATP. Then, light has also struck photosystem 1, and a pair of electrons have been lost. These electrons, along with protons, join NADP, and this means it becomes reduced NADP. I know that even though it's added the electrons become reduced, but that's what happens. So electrons, along with protons, join NADP, making it reduced NADP. The electrons from the oxidized photosystem 2 replace the electrons lost from photosystem 1, so continue. And finally, protons from the photolysized water take part in chemiosmosis to make ATP, and then they're captured by NADP in the stroma. They will be used in the light-independent stage. So we've basically just gone over the whole of the light-dependent stage. Fantastic! The main things we went over the role of water, photophosphorylation, cyclic phosphorylation, and non-cyclic phosphorylation. Cyclic photophosphorylation and non-cyclic photophosphorylation. It's confusing, the words all sound the same. If you want a quick definition of photophosphorylation, you can say that it's the making of ATP from ADP and PI in the presence of light. So great. Do you want some questions to make sure you know it? You could outline the role of water in photosynthesis. Also explain how the light causes stomata to open and why is that significant. And the horrible suggest question, you could do suggest why a lack of iron in soil can reduce growth in plants. That's what I'm going to go and do right now because I hate the suggest questions. So, yep, that is all of the light independent stage. Fantastic, and super good luck with your exams in January because they're coming out really soon. Or you might have them in June, whenever, but mine's in January and it's really soon. So, good luck with your revision.